Hi guys, Anna is here, and in this video we're going to be talking about the process of translation and everything you need to know for your biology A-level. Grab a piece of paper so we can work together, and without further ado, let's jump into it. So first of all, I want you to start with an overview of what we covered so far in my previous videos, which you can click on the links above. So let's look at what we've covered so far. So let's draw a circle and a big nucleus. So we're looking specifically again at eukaryotic cells. And the first process that we've covered so far is transcription. So how DNA is transcribed into messenger RNA, which happens in the nucleus. Then the second process we covered is splicing. So cutting out of the non-coding sections of pre-mRNA, which are called introns, and then making it into mature mRNA. And then mRNA will leave the nucleus and it will travel to the ribosome where the translation will occur. So let's just label it over here. Okay, so this is, guys, what we covered so far: transcription, splicing, and in this video we're going to talk about translation. But before we can discuss translation as an overall process, we need to look at the structure of mRNA first. So I want you to draw it like as this rectangular. Remember, it's still single-stranded, but it's just easier to represent it this way. So remember, guys, that mRNA is made of many codons. And a codon is a sequence of 3 bases within mRNA, which code for a single amino acid. So 3 bases code for 1 amino acid. So mRNA will be made of many codons, but the mRNAs will always have start codon at the beginning, which is always AUG. Then we're going to have many different codons in the middle, and then at the end there will be a stop codon. And stop codon basically doesn't code for any amino acid, and it basically is a symbol used by ribosome to know that, okay, the translation needs to be terminated. Very similar, right? So to the structure of the gene. So we have the start codon that marks the beginning of mRNA and stop codon that marks the end of mRNA. So, right, guys. So remember, will be a very good place to make a flow chart here. So DNA base order is important and will be coding for a codon order. Codon order will then code for amino acid order, which will code for protein. And just have that as an overview. Okay, so you might have noticed that I already mentioned the word genetic code. And basically, genetic code is a series of codons that code for a particular sequence of amino acids and eventually a protein. So it's basically the information that is contained in DNA and passed to make a protein. And for A-level, you need to know several features of genetic code. And so just remember, when we talk about genetic code, it's about series of codons. The first one is degenerate. So genetic code is degenerate. What it means, guys, is that basically several codons can code for one amino acid. So let's take leucine as an example. And if you kind of look at the table of the codons and their sequences, you will notice that CUU, CUC, CUA, CUG, UUA, and UUG will all code for one amino acid, which is leucine. So if there is a mutation from U to C, it doesn't matter because it will still code for the same leucine, which is an example of a silent mutation. Okay, let's jump to the second feature of the genetic code that is called universal. And in this case, guys, it means that same codons from one organism code for the same amino acids. So what it means, guys, if we were to take and isolate an insulin gene from a human cell, and we were to then insert it into the bacterial cell, then bacteria cell would transcribe and translate insulin gene, and it would produce human insulin protein, which will be part of your course in year 13, and I'll be making further videos on that. So that means universal. And the third final feature of genetic code that you need to know is called non-overlapping. And non-overlapping means that 3 bases code for one amino acid. There is no overlap between them, and each base is used once per codon. So let's just draw another example. So let's just draw a sequence of codons AUG, CUU, and GAA. And so the cell will read it first. So AUG will be read first and produce an amino acid, then CUU, and then GAA. And then there will be no overlap at all between the three of them. So far we've covered the structure of mRNA and we discussed the features of the genetic code. But there is a second component that is required for translation, and that is tRNA, which stands for transfer RNA. And you guys are required to know the structure of that. So what we're going to draw it as a clover leaf shape, like this. Just leave a little bit of space at the 3'. It's still single-stranded, but there are a couple of features that you need to know about that. Okay, so first of all, the function of tRNA is to bring a specific amino acid to the ribosome. So how does that happen? So at the bottom of the tRNA, there will be a sequence of 3 bases that is called anticodon. The second feature that you need to know is hydrogen bonding, which is, funnily enough, although tRNA is single-stranded, the way it falls on itself, it can have hydrogen bonding in some parts of it. And I've seen this as an example question, so you guys need to know that hydrogen bonding is present in the tRNA. And the last side that you need to know is amino acid binding side. And this is where the amino acid will attach in the tRNA. And this specific amino acid binding site will correlate to the sequence of anticodon. So let's draw mRNA, and basically let's say that anticodon is going to be hypothetically GAA. So that means that this anticodon sequence will be complementary to a codon on mRNA, so it will be CUU in this case. And in exam, guys, you need to use very specific language. You need to say that anticodon on tRNA is complementary to the codon on mRNA. And I've seen basically if you just say codon, anticodon, they don't count it sometimes as marks. So just kind of keep that in mind as an exam hint. So only 3 structures that you need to know is hydrogen bonding, anticodon, and amino binding site. Okay? And this is the overall structure of tRNA. So we finally are at the stage, guys, to understand how translation happens within the cells. And we need to draw a ribosome. Ribosome is an organelle found in the cytoplasm that in prokaryotes is kind of just floating in the cytoplasm. In eukaryotes, it will be attached to the rough endoplasmic reticulum. As a reminder, we find there are 70S ribosomes in bacteria and 80S in eukaryotic cells. So the ribosome consists of a large subunit and small subunit. I always call it like a burger bun, easy to remember. And then mRNA will kind of bind to small subunit, and it will sit in between both of those subunits. What is commonly missed in talking about translation is that actually the large subunit has 3 binding sites, which are called APE. You don't need to know the names of those, but just appreciate that they're there for now. And what you need to know is that there are only 2 tRNAs can bind to the ribosome at a specific time. So let's draw them in. So the tRNAs, I always simplify them by this stage. So obviously it's just easier to draw them, but the structure is as they are on the previous page. And I just draw them kind of with those little amino acids attached at the top and then the anticodons at the bottom. And the anticodons on tRNAs are complementary to codons on mRNA. So only 2 tRNAs can bind to ribosome at the time. They bring the amino acids and then they will exit through the E site. And as they exit, they will leave the amino acids behind. And as you see, guys, it's difficult to visualize it in 2D. So I recommend watching some 3D animations and how that happens. But basically, as they bring in 2 amino acids, the polypeptide chain will grow and there will be peptide bonds between the amino acids formed. And basically, the idea is that the ribosome will move on the mRNA as a conveyor belt, and basically tRNAs will come in and bring the amino acids which are complementary to the codon. And this is the overall summary of everything you need to know for translation for your A-Level Biology course. Please, if you like this video, press the thumbs up subscribe to my channel, and please do let me know what next videos you would like to see in the comments below, and I'll see you in my next video. Goodbye.